So now we are going to start a discussion on detail about what type of oxidative or aromatic hydroxylation conversions are done for the metabolism of an arene ring containing or if the presence of electron donating group it is there in the aromatic ring. Suppose, now what are the examples of electron donating groups? Nitro group is electron donating, aldehyde group is electron donating, amine group is electron donating. So if there is substitution of the electron donating group in this arene ring, so it is go for a metabolism through aromatic hydroxylation. So various examples we have discussed, that is an amphetamine molecule metabolized through or biotransformed through process of hydroxylation, then a phenylbutazone, phenytoin. So various drug molecules are metabolized, or particularly this ring structure of various drug molecules are biotransformed, are made easily available for metabolism through hydroxylation reaction. Now, a next type of aromatic hydroxylation examples, if see earlier case we have discussed if the arene ring is substituted with electron donating groups. Now if the arene ring is substituted with electron withdrawing groups like chlorine, so halogen atoms, so that time what type of hydroxylation will happen? Now in case of earlier case, the hydroxylation process is fast due to the presence of electron donating group here in the structure. But in the case of arene oxidation, if the arene ring is substituted with electron withdrawing groups like halogens, it will slow down or resist for hydroxylation. So means in simple language मध्ये आपल्याला काय म्हणता येईल, जर इलेक्ट्रॉन विड्रॉइंग ग्रुप्स लाईक हॅलोजन्स वगैरे प्रेझेंट असतील त्या रिंग मध्ये, सो इट विल स्लो डाऊन और इट विल रेजिस्ट द aromatic oxidation process, that is aromatic hydroxylation process. Now such type of example of drug molecule is clonidine hydrochloride. See, the benzene ring is there, but it is attached to the electron withdrawing groups. Now, if the ring is attached to the electron withdrawing groups, like halogen atoms, it will slows down the process of aromatic hydroxylation. So, such type of examples are clonidine hydrochloride, where the electron withdrawing group can be a chlorine. Now such example is probenicid. So such type of metabolic reactions either slows down or resist for the aromatic hydroxylation. This is there. Now the next example of aromatic hydroxylation procedure, or the next statement, next conversion, if the compound contain multiple aromatic rings. See, आतापर्यंत आपण कसले एक्झाम्पल्स बघितले, इफ द कंपाउंड इज अंडरगोइंग aromatic hydroxylation, presence of arene ring, but it is one or two and it is go for easily for aromatic hydroxylation. Then we have seen the examples if the aromatic ring is attached to the electron withdrawing group, faster is the aromatic hydroxylation. Then we have seen the case number 3, if the arene ring is substituted with electron withdrawing groups, so it is going to slows down the hydroxylation process. Now case number 4 is if the compound contain more than one arene ring, multiple aromatic rings, so what happen in that case? Hydroxylation is preferred. Generally hydroxylation कुठल्या cycle वरती किंवा कुठल्या ring वरती होतं, so the hydroxylation is preferred on which ring? The ring which is rich in electron. See, so If more than one or multiple aromatic rings are present in any drug molecule, so the metabolism of that type of drug molecule is go for, or it is done via what type of metabolism reaction? So किंवा कुठली रिंग फर्स्ट ऑफ ऑल गो होते किंवा फर्स्ट ऑफ ऑल रिएक्ट होते और फर्स्ट गो फॉर हाइड्रोक्सिलेशन रिएक्शन? The ring which is preferred on the electron rich ring. See here. Now we are going to take an example of a para-chlorobiphenyl. So the condition is more than one aromatic rings are present, but the one ring is attached with the electron withdrawing group. Now we know if the ring is substituted or attached to the electron withdrawing group, it is going to slows down the hydroxylation procedure. So या case मध्ये or in this case, which ring is go first or which ring is preferred for the aromatic hydroxylation? Ultimately or obviously, this ring is preferred for the aromatic hydroxylation type of reaction. So the examples of such type of aromatic hydroxylation is para-chlorobiphenyl. Now, the next one is chloropromazine. The drug molecule which is used as an treatment of psychotic disorder, that is chloropromazine. Now, what happened in the case of chloropromazine? Chloropromazine drug molecule also have a presence of multiple aromatic rings. Now, but out of this, which ring is rich in electron? Now we know this presence of double bond is nothing but continuous pi cloud of electron. So this ring is rich in electron. See, if we are talking about this middle ring, so there is only 4 pi electrons are present. If we are talking about this last ring, so the last ring is attached to the electron withdrawing group. So in this case, which ring is go first or prioritized for the aromatic hydroxylation? Kiva metabolism of such drug molecules are started with the hydroxylation of this aromatic ring. So these are some of the case studies or the examples of the drug molecule if the compound contain multiple aromatic rings for the hydroxylation reaction. Now the last example or the next example is if the presence of ring atoms are different. See, one arene ring, another one is a 7-membered ring, azine, and the last one will be the same arene ring which is substituted with electron withdrawing group. So same is the case with the first drug molecule. If electron withdrawing group is present, it is going to slows down or resist the aromatic hydroxylation procedure. So which ring is preferred for the aromatic hydroxylation? Ultimately and but obvious, this ring is go for metabolism on priority basis for what type of reaction? Aromatic hydroxylation reaction. So or this ring is first of all go for metabolism as compared to the another ring for what type of reaction? Aromatic hydroxylation reaction. So they may ask the question or they may give the structure and ask you to draw the first metabolic reaction. For 4 marks or 3 marks, this type of questions are there. The structure is in front of you. Only you have to select which ring is go for first type of aromatic hydroxylation reactions. So the 4 marks are there in your pocket. Is it clear to everybody? Now, the next type of reaction is under the case only phase 1 reaction, that is aromatic hydroxylation. Second case study is if, see, polychlorinated biphenyl ring structures are there in the drug molecule. See, we have seen an example of presence of electron donating groups. Then if the one of the ring is substituted with the electron donating groups. So these are there. Now, if this is the case, that is, arene rings are substituted with the more electron donating groups. So in this case, body generally resists or slows down the procedure of metabolism, and that's why this compound is coming under the category of environmental pollutant, because it is not at all metabolized, or it will slows down the metabolism procedure. That's why it is going to create the toxicity and health hazards to the living organisms. So these are the cases of types of oxidation reactions. Now, the next case study is oxidation of aromatic moieties, that is via aromatic hydroxylation, and the reactions are proceed in different type of enzyme examples or different type of drug molecules, how they come acting on aromatic ring. See, if the aromatic ring is there, so if it is going, first of all, for the Kiva, what are the different ways by which the aromatic ring is go for aromatic hydroxylation reaction? आतापर्यंत आपण काय बघितलं, कुठली रिंग aromatic hydroxylation reaction ला prefer केली जाते. But we have seen in second slide number 2 only, there are different type of enzymes which will convert the drug molecule into different forms. Now, similar case is here. The drug molecule is exposed to different type of environment, different type of enzymes, different type of biochemical entities. So what happened to the drug molecule if it is going to react with the different type of biochemical entities? So first reaction is the aromatic rings, no doubt, if it is going for hydroxylation reaction, it is proceed via intermediate formation of compound, that is arene oxide. Now what happened to this arene oxide if it is exposed to? First of all, it is ultimately or spontaneously going to rearrange its structure. If the arene oxide molecule is going to rearrange its structure through spontaneous rearrangement, so it is going to shift his hydroxyl group or his oxygen and hydrogen group to the different position. See, the structure of arene oxide is in this manner. Arene oxide means formation of one more oxygen bonding in the presence of double bond, carbon-carbon double bond. So this arene oxide will be change or rearrange its structure in this way, so that oxygen is going to form the double bond and hydrogen atom is shifted to the next carbon atom. This type of shifting of oxygen and hydrogen is known as Nih shift. Nih shift means 1 to oxygen hydrogen shift. So this type of shift will go further for the aromatic hydroxylation reaction to forms arenol. This particular reaction is to the large extent is reverse, to the small extent is irreversible in nature, but generally they are going to form the arenol as a metabolic product. So this is if the arene oxide is going to form or is going under the spontaneous rearrangement type of reaction. Now, the next metabolic pathway, when the arene oxide is exposed to the water molecule. Now, we all know our body is going to be have presence of 98% of water, 78% of water. So, this arene oxide in the presence of water molecule, it is hydroxylated and going to form the trans dihydrodiols. So when it is exposed to the water molecule, it is going to form the dihydrodiols, substitution of the hydroxyl group at two different positions, because this and this both carbon atoms are exposed to the hydroxylation procedure due to the presence of this arene oxide type of compound. Now the third type of way through which the aromatic hydroxylation takes place is glutathione adduct. So the glutathione molecule is going to be react with the arene oxide molecule, and they are going to form one intermediate product, that is glutathione adduct, before aromatic hydroxylation. Now these three type of reaction, that is rearrangement into an arenone, then formation of trans 2 diols, trans dihydrodiols, and formation of glutathione adducts, they are detoxified and eliminated from the body. See, drug ने त्याची action दिली आहे, it is going to be metabolized, and the remaining amount or the unwanted metabolic products are detoxified and eliminated from our body in this way, that is through spontaneous rearrangement, reacting with the water molecule, and forming an adduct with the glutathione molecule. Now the next is a macromolecules. Macromolecule adducts, that means the arene oxide is going to form a bond with the macromolecule. Macro means big molecule. Another big molecule it is going to be reacted with. So there is formation of macromolecular adduct, which is so there will be an formation of covalent bond with the macromolecule and the arene oxide. So the examples of the macromolecules are DNAs, RNAs, and protein. We have seen that the drug molecules are bind to the protein molecules, so it is distributed, easily distributed, but unable to excrete out. It is deposited in our body. So this procedure leads to the cellular damage. So these are the various types of aromatic hydroxylation steps when it is undergoing or when it is exposed to the different type of products or different type of enzymes, and they may carry out the metabolic reactions. Now the next is a oxidation of olefins. Now we have seen, up till now we have discussed the oxidation of aromatic hydroxylation reaction, aromatic rings. Now we are going to discuss the different type of or second type of oxidation reaction under the phase 1 oxidative step is if the drug molecule contain alkenes in its structure. आतापर्यंत आपण कुणाचं ऑक्सिडेशन बघितलं, या बेन्झीन टाइप ऑफ रिंगचं ऑक्सिडेशन कसं होतं किंवा ती रिंग पर्टिक्युलरली कशी मेटाबोलाइज केली जाते. सो हाऊ इट इज मेटाबोलाइज्ड? It is metabolized through formation of arenol and excreted out throughout the body. Now we are discussing the oxidation of olefins. So the metabolic oxidation of olefins. The olefins is nothing but carbon-carbon double bond. It will lead to the corresponding epoxide. So in this case also, the double bond, that is olefin, is going to form the epoxide in its structure, and that epoxide is known as oxirane. Now these epoxides are generally more stable and more water soluble. So these epoxides, when the double bond is converted into in its epoxide, they are more stable and more water soluble than the arene epoxides formed from the carboxylic compound. So this type of epoxides are more soluble in the water and more stable than the epoxides which are formed by these aromatic rings. See, these aromatic rings are also going to form the epoxides, and this double bond, carbon-carbon double bond or olefin in the structure is also going to form the epoxide. But this type of epoxides are more stable than the aromatic epoxides when we have compared the stability point. So such type of examples, when a drug molecule, that is carbamazepine, is undergoing what type of oxidation? Oxidation of olefins, that is oxidation of carbon-carbon double bond. That means metabolism of this ring. So that time there is formation of epoxide in nature. So that formed epoxide is known as oxirane. So the drug molecule which is undergoing this type of reaction is known as carbamazepine, antipsychotic drug, 10-11 epoxide. And after this formation of oxirane epoxide, later on it is easily hydroxylated and metabolized into trans 1011 dihydroxycarbamazepine. So the carbamazepine drug molecule is metabolized into trans 1011 dihydroxycarbamazepine. Now the next drug molecule is oxidation of next examples of such type of drug molecules are metabolic oxidation of olefin leads to the corresponding formation of epoxides and they are more water soluble. So such type of examples are benzopyrene. It will convert it into in its epoxide, that is benzopyrene 7,8 epoxide, and it is converted into its particularly hydroxylated derivative, that is 7R 8R 7,8 dihydrobenzo tetraphene. Now, so the second type of metabolism किंवा second type of hydroxylation reaction is oxidation at benzylic carbonate. Now the next one will be oxidation at carbonatum attached to the aromatic rings are susceptible to the oxidation. So the carbonatum, when it is going to the attached to the aromatic ring, see this type of situation when any carbonatum, see this R carbonatum or R is any alkyl group, when it is attached to the aromatic ring, so this type of carbonatoms we can call them as a benzylic carbonatoms. They are susceptible to the oxidation and from corresponding alcohols. So this is the carbonatum which is attached to the aromatic ring. So it will easily undergo an hydroxylation reaction to form alcohols or corresponding alcohols. They will undergo, if primary carbonatum is there, then primary alcohol, oxidize further to the aldehydes and carboxylic acids. If the carbonatum is secondary in nature, it will convert it into the ketone derivatives, secondary alcohols, आणि secondary alcohols further oxidation झालं की काय तयार होतात? Ketones, and the corresponding alcohol may be conjugate with the glucuronic acid in phase 2 reaction. This is all about phase 1 reaction, and if it is bonded to the if it is conjugated to the glucuronic acid, it will enter in the phase 2 reaction. Now, examples of such a type of oxidation reactions are when the carbon atom is attached to the aromatic ring. See, this is the carbon atom. This is the carbon atom which is attached to this aromatic ring. So, how come this carbon atom is hydroxylated? Kyunwa metabolized? So, this benzylic position carbon atom are susceptible to the oxidation and they are going to form the corresponding alcohol in first metabolic reaction. So, the example of such drug molecule is tolbutamide, which is used as an anti-diabetic agent for the treatment of diabetes. म्हणजेच आपल्या बॉडीमध्ये टोलब्युटामाइडचं मेटाबॉलिझम किंवा एक्सेस अमाउंटचा टोलब्युटामाइड कसा बॉडीमधून बाहेर टाकला जातो with the help of, after its treatment, with the help of metabolic reaction. आणि कसल्या टाइपचं मेटाबॉलिझम होतोय? Now this aromatic ring is going for oxidation and the carbon atom which is attached to this aromatic ring is go for oxidation to form the corresponding alcohol. So this methyl functional group is going to form the methanol or methyl alcohol. So this alcohol functional group, as it is primary alcohol, it is going to be formed into an alcohol metabolite, and this alcohol metabolite is further oxidized into an aldehyde. Aldehyde further oxidized into the acid. So there is formation of an carboxylic acid metabolite. This carboxylic acid metabolite will be removed out from our body now through excretion. So this is a carboxylic acid metabolite for with respect to the tolbutamide. There are drug molecules, that is tolmetine, which is also metabolized in the similar way because it is also attached to the methyl functional group. Or there is also one more methyl functional group which will be attached to the tolmetine. So this is also going to be proceed with the same mechanism of metabolism in order to form its carboxylic acid derivative. Now, so in this way, the benzylic carbon atom, which is attached to the any arene ring, is metabolized in our body. Now, the next case is oxidation of allelic carbon atom. So, if the carbon atom is allelic in nature, see here the carbon atom is bonded to the another two carbon atoms, but the carbon atom is outside the ring. आतापर्यंत आपण काय बघितलं, मिथाइल फंक्शनल ग्रुप सिंगल कार्बन ॲटम असेल तर, पण जर तो कार्बन ॲटम इज अलिलिक कार्बन ॲटम असेल, then it is undergoing hydroxylation reaction not by the regular hydroxylation. Such type of compounds are undergo microsomal hydroxylation. So some microsomal hydroxylation reactions takes place in our body. So this type of carbon atoms or the drug molecules of this type will go for microsomal hydroxylation at this allelic carbon atom. So which is observed in commonly in the drug metabolism of what type of drug molecules? Quinidine drug molecule. So this quinidine is metabolized in our body with the help of microsomal hydroxylation reactions to form a metabolite that is 3-hydroxyquinidine. So it is easy to excrete out from our body. Now the next example of microsomal hydroxylation type of reaction is allelic carbon atom, which is commonly observed in the drug metabolism. So such type of drug molecules are hexobarbital. Barbituric acid is used as an sedative and hypnotic. So this sedative and hypnotic drug, that is hexobarbital, is metabolized. That drug molecules आपल्या body मध्ये metabolism कशा पद्धतीने होत आहे? It is metabolized through hydroxylation of this arene ring to form 3-hydroxyhexobarbital, and after that 3-hydroxyhydrobarbital is go for either glucuronide conjugation, that means it is transported to the phase 2 reactions, or it is converted into 3-oxohexobarbital. So hydroxyl functional group is converted into ketone functional group, that is 3-oxohexobarbital. So likewise, this type of reactions are proceed further for the metabolism of various drug molecules. So the conditions and situations when we have discussed about types of phase 1 reaction is, number 1, very important one will be aromatic hydroxylation. Then aromatic hydroxylation if presence of electron withdrawing group, aromatic hydroxylation presence of electron donating group, then aromatic hydroxylation if the carbon atom is outside the ring, aromatic hydroxylation if the carbon atom is double bonded to the another molecule. So various type of metabolic reactions we have discussed today.